I'll be covering block objective 40, which is the cellular and subcellular components of primary immunodeficiency. So in essence, it's the molecular basis of primary immunodeficiencies. Let's start off with a question. It says a child is known to have a primary immunodeficiency. He presented to your care with an extremely severe case of infectious mononucleosis. His past medical history is not significant for any other bacterial or viral infections besides Epstein-Barr virus. What is the mutation that is most likely associated with this disease? So we have Wasp, IL-2 gamma chain, BTK, SAP, and C1INH. You go ahead and pause it, and I'm going to show you the answer right now. The answer is SAP, or SLAM-associated protein. SLAM-associated protein is associated only with an increased risk of infection with Epstein-Barr virus. It's in X-linked lymphoproliferative syndrome, and we're going to go through the rest of this presentation. We'll basically explain why the others are not the answer and why this one is. What am I not covering? I don't think I'm going to have time to cover complement, so it's not going to be in here. And there's about 1 million immunodeficiencies that I'm not going to cover. But I am going to cover the ones that I think are high yield for step 1. And so here we go. Alright, you can get these tables in my study guide. It's basically a breakdown of like the simplest possible explanation I could make for each of these diseases. And I'm not actually going to cover the Mendalian susceptibility to mycobacterial diseases, but you have that right here as far as the super small, fast breakdown. Starting off with innate immunity, and then we'll go into the adaptive immunity, and then we'll go into systemic immune problems. Alright, first is the chronic granulomatous disease. So I remind you, what is a granuloma? A granuloma is basically where you have a pathogen and it's not being able to be destroyed. It's fighting off all of your neutrophils, it's fighting off all of your other phagocytes. So what happens is the macrophages come in and build a wall around the infection so that it can't spread, essentially. This is a granuloma, really common with tuberculosis. It's common with certain fungal infections, but it's not common with all the other bacteria. Usually your body has no problems fighting off things like we'll say staph or things like Pseudomonas. Your body doesn't even need the T cells to do that. Your neutrophils can go in and destroy some staph infection real quick. But when there's a defect in the oxidative burst, the neutrophils have a problem. They can't kill the bacteria. So we're going to explore really quick what is the oxidative burst. So I got my light microscope right here, and I'm going to go ahead and put a plate of cells on. And I turn on the light, and now we're going to zoom in to the high power focus. Just going to keep on going in until there's a cell. Let's zoom into the cell and see what's going on inside of there. There it is. We have the oxidative burst. And so let's take a look and see what's happening. What we have is we got oxygen right here, and by the enzyme NADPH oxidase, using NADPH, oxidizing the NADPH, we are turning oxygen into superoxide, which is basically we're giving it an extra electron. Then we're using superoxide dismutase, that with water we can turn superoxide into peroxide, hydrogen peroxide. And then with myeloperoxidase can turn hydrogen peroxide into bleach by adding chlorine. This will then go out and find all the microbes anywhere and everywhere and kill them all. So this will kill every microbe. Well, maybe not every microbe, but it'll kill a lot of them. Now, just as a side note, this hydrogen peroxide, it can actually go through a couple of different pathways. It can be turned into the bleach, like we said, but just as a as a biochemistry tie-in, remember that H2O2, if we get too much of it in ourselves, we use catalase, and we turn it into H2O. You can turn a couple molecules of peroxide into H2O and O2. Or And it seems like probably just as often as catalase, our bodies will use glutathione to do the same thing. So glutathione, which is this SH stands for the thiol group. The sulfhydryl group is the thiol group. It will take two glutathione molecules, and it can convert those into the oxidized glutathione, allowing the hydrogen peroxide to be turned into water. Then to get the oxidized glutathione back to the reduced form, what you would do is you would use, again, NADPH. NADPH would be converted into NADP, and now we have two different things that are using up NADPH, and we've got to get the NADPH back. And so how do we get that back? We use glucose 6-phosphate dehydrogenase. So I have glucose 6-phosphate. It will interact with NADP, reforming NADPH by the enzyme glucose 6-phosphate dehydrogenase, G6PD, and that will form 6-phosphoglycerate. So that's just a side tie-in, but the big important part, the oxidative burst, is this part right here, and we see that we can get rid of H2O2 with catalase. That'll be important in a minute. But if we were to inspect this enzyme, NADPH oxidase, we would see that it's made up of 6 subunits. So here is my NADPH oxidase. So I'm just going to put NOx. And what it has is 6 subunits. And one of those subunits is a GTPase. So it can use GTP for energy if it needs to. Now the rest of these are the FOX enzymes. These are the FOX enzymes. So FOX stands for phagocyte oxidase. So FOX, we got FOX91, we've got FOX67, we've got FOX phagocyte oxidase 47, we've got 40, and we've got 22. Now this particular phagocyte oxidase, this particular one is found on the X chromosome. These two are found on autosomes. And any of these 3 phagocyte oxidase, any of these FOX proteins can have a defect in a mutation in them that will inactivate NADPH oxidase. So with FOX91, we can get an X-linked recessive, and with the other 67 and 47, we can get an autosomal recessive defect in the oxidative burst. This defect in the oxidative burst will cause, the defect prevents us from forming superoxide. So oxygen, superoxide, peroxide, bleach. And what does that cause? That causes us to have a defect in killing for all catalase-positive bacteria. So what are the catalase-positive bacteria? It's the mnemonic bin places. The bin, I keep that there because I think the first aid gives you the mnemonic need places, and then the only thing that means anything is the N for nocardia. But there's also Burkholderia that's catalase positive, nocardia, Pseudomonas carpaceae. There's a whole bunch, so just remember all the catalase positive. When you finish it up, it's Listeria, Aspergillus, Candida, Enterobacteriaceae, and then the 2 different staph species. Or you could say Staph aureus and Serratia. All of these produce catalase. Now, you're thinking right now, if you don't have hydrogen peroxide, if you don't have superoxide, you didn't make any peroxide, so why would catalase have any impact on these guys at all? And the answer is that bacteria, just in general, also make a small amount of H2O, and so they can feed into myeloperoxidase to form bleach. But the catalase positive bacteria are destroying all of the peroxide that they are producing before they send anything out. So they're not sending out any peroxide, but the catalase negative bacteria will, and it'll feed in and you can make bleach with it. Now, there are also deficiencies in myeloperoxidase that can have similar effects, but again, the H2O or H2O2, the peroxide by itself can kill some bacteria. So the exact bacteria that you're going to affect with a myeloperoxidase deficiency is going to be a little bit different. But in general, the NADPH oxidase deficiency is the most common. So we have 3 leukocyte adhesion deficiencies, so LAD, leukocyte adhesion deficiency 1, 2, and 3, and it's basically pretty simple. If you remember how the leukocyte adhesion molecules work, so here I have my blood vessel. I've got all my cells, my endothelial cells in the blood vessel, and whenever we undergo inflammation, what happens is the blood vessel dilates. So back here in this area, the blood was flowing through. In sort of a high stress, a high shear stress type of flow, and so you have it going really fast, and you have the fastest movement here in the center. You have friction against the wall. The friction is causing shear stress, so it's ripping off any cell that gets close to the cell wall is being ripped off by the shear stress and sort of pulled to the middle. But whenever the blood vessel opens up, the amount of that shear stress, the amount of flow speed in the center becomes very close to the same as the flow speed on the side, and so it allows some of the leukocytes to move toward the outside. So you'll remember that the steps of a blood cell, of a leukocyte, binding to the endothelial cell and moving into the site of infection is basically 4 steps. You've got margination, rolling, adhesion, and transmigration. You could also add a 5th step in there of just the chemotaxis to the actual site of injury. Now the margination step I just explained, it's pure physics, it's hydrophysics. Now the next step, we're going to zoom in and take another look. We have these surface molecules on these endothelial cells called selectins. So you have P-selectin, E-selectin, and things that cause that. So histamine release, histamine and thromboxane A2 will both cause the expression, or not the expression, because these selectins are inside in endosomal packages, and whenever histamine is acting on these cells, it causes them to move towards the surface. So they come out toward the surface with histamine and thromboxane A2. If you get IL-1, IL-1 will actually go in and cause nuclear signaling to upregulate the expression of selectins. And it's important to remember, like when we're talking about selectins, P-selectin specifically, they're contained within the Weibel-Palade bodies. And then I have my leukocyte up here. My leukocyte has this protein on it, and on that protein it has a whole bunch of sugar moieties. And these sugar moieties have a little piece on them called sialyl Lewis X. So we've got sialyl Lewis X on these, and these sialyl Lewis X sugar moieties are going to bind to the selectins. So if you remember that lectin is a sugar, and we are binding selectins to sugar moieties. That's kind of an easier way to remember it. And remember that this interaction between selectin and sialyl Lewis X, this is a lower affinity, so it doesn't trap the leukocyte. So here's my leukocyte again. It doesn't trap the leukocyte, it just allows the leukocyte to slowly kind of roll, like a Velcro ball would roll down a Velcro wall. I suppose, I guess if the ball was heavy enough, it would just kind of stick to it and roll down the wall. Now if we go over to the next endothelial cell, this one is expressing something called a CAM, a cell adhesion molecule. And there are different types of CAMs, I'm not going to go into that. And this time my leukocyte is expressing something called an integrin. And when that integrin binds, it does two things. The first thing it does is it stops it. It's a high affinity. That's the word I'm looking for. High affinity adhesion, and so it stops it. The second thing it does is it sends a signal down to the nucleus, or down, it sends intracellular signaling to say, hey, I'm bound, this is where I need to be, it's time to go across. So signaling goes down and says, hey, it's time to go transmigrate. So let's look, and now that we have that under the belt, the integrins, they are made up of alpha chains and they're made up of beta chains. So you can have something like alpha 1, beta 1, and that's a specific type of integrin. If I remember right, that integrin specifically is LFA1. I could be wrong. Not really a big deal, not the point for this. The point is that different cells produce different integrins, and one that we're looking at is an integrin that specifically has beta 2 as its chain. And the beta 2 chain on leukocytes, whenever it gets mutated, it can't bind efficiently, so you get the margination, you get the rolling, you just don't get adhesion. So it's a defect in adhesion. With type 2 leukocyte adhesion deficiency, there's a defect in the protein GDP fucose, and it's the job of that. So inside of the leukocyte, inside of the Golgi apparatus, so here's my nucleus, here's my ER, and then I have my Golgi apparatus. Inside of that Golgi apparatus, you have to import fucose, and fucose gets added to and incorporated in the Sial-Lewis X, and whenever it doesn't happen, you get defects in Sial-Lewis X, and with that, you have problems with rolling. So it's a defect in rolling. And then LAD3, there's a defect in something called Kindlin3, and this is basically a signaling molecule for the integrin to say, hey, we're here, let's go in. And so it's sort of the step between adhesion and transmigration. So you get a phenotype that's similar to LAD1 plus a mix of Glanzmann thrombasthenia. I'm not going to go through what Glanzmann thrombasthenia is, except to remind you that it is a defect in the GP2B3A receptor, and that's for platelet aggregation. So you get a combination of two phenotypes with this LAD3 deficiency, and it's due to a defect in Kindlin3. The main thing that you're going to get with all of these is recurrent bacterial and fungal infections, usually around the skin. Okay, the next immunodeficiency is Chiari-Kakashi syndrome, and this is a defect in the LST protein. LST, it stands for some lysosomal protein. Basically, it's lysosomal trafficking. So lysosomes have to be moved around in the cell. They have to be fused with other endosomal membrane-bound things, and so whenever you can't do that, you can't kill stuff. So there's two ways to kill a bacteria. As far as an extracellular bacteria, there's two ways to kill it. You can either phagocytize it, so you're going to phagocytize it, and then once you do that, you can either do the oxidative burst, or you can merge it with a lysosome. And when they get merged with the lysosome, if the lysosomal trafficking isn't working properly, then you have these giant lysosomes that you see inside of neutrophils. And then lysosome-like, I put lysosomally-like, I don't know why I put the Y on there, lysosomal-like endosomes. So other endosomes that are similar in their structure to lysosomes, they also have problems. One of these specifically is the melanosome. The melanosome is you have in your skin, you have mostly cells that are making skin. You have epidermis that's making keratinocytes. These keratinocytes are for the skin. But you also have these random melanocytes, and these melanocytes are handing off melanin to like 25 or 30 different keratinocytes. And this melanin is stored inside the melanin is stored within the melanocyte inside of a melanosome, and that can't be trafficked properly. It can't be handed off to other cells. So what you see with this is you see neutrophils that have giant lysosomal complexes during their maturation. So whenever a cell's maturing, it has to build stuff up and it has to break it down. Some of that breakdown happens inside of the lysosome. Some of it happens with ubiquitin. Some of it happens in lysosome. It happens all over the place. But you get these giant lysosome complexes, and because they can't be trafficked, they can't be moved where they need to be moved. And the neutrophil will typically die during maturation. Some of them will make it to maturity, but you can have a mild neutropenia due to this disease. Like I said, the melanosomes cannot distribute melanin, and then you have lysosomal trafficking problems in the neurons that usually cause various types of neuropathies and defects within the neurons. So the problem with the immune part is that you have increased in pyogenic infections. You're going to get albinism because you can't hand off the melanin, and you'll get neuropathies. Okay, with natural killer cells, you can have a defect in GATA2. This is a transcription factor. It will actually impact more than just natural killer cells, but it has its largest impact on the natural killer cells. But you can also get a defect in MCM4, which is a specific type of DNA helicase, and it's only going to be seen as a loss of natural killer cells. Then also with CD16, the mechanism is not exactly known. Okay, so toll-like receptors. Let's go back to block 4 with immunology. We have several different toll-like receptors, and those toll-like receptors are attached to a molecule called MyD88. MyD88 will send a signal to a complex of IRAC molecules, and specifically we're looking at defects in IRAC4, so I'm going to put that there, but it's really a complex of a bunch of IRAC, and then those will go downstream and activate NF-kappa B, NF nuclear factor kappa B. Then nuclear factor kappa B goes and activates the DNA and causes it to produce cytokines. So you get cytokines. There are several different defects in TLR signaling. The most common are defects in MyD88 and IRAC4. You can also have defects in TLR3 specifically, but it uses let me back up. You can have a defect in other proteins that will specifically affect TLR3, and those proteins are the TRIF and ERF pathway to NF-kappa B. So TRIF and ERF is what TLR3 uses as an alternate signaling to get to NF-kappa B. And whenever you have defects in TRIF and ERF, you have higher susceptibility to herpes simplex encephalitis. And the phenotype with this is going to be anhydrotic ectodermal dysplasia plus immunodeficiency. And the immunodeficiency, like I showed you, is going to depend on which signaling molecules we have affected. I have no idea what this means, but and I don't think it's high yield either. But I would say you do get that as the phenotype in TLR signaling, so I'll probably end up looking it up. So that finished up our innate immune system. Now we're going to start on adaptive, and we're going to start first with the several different kinds of SCID, severe combined immunodeficiency. This is Bubble Boy. So what I think a lot of people that have looked at the immunodeficiencies don't realize is that there are more than one type of SCID. So you can have X-linked SCID, which is what most people think of. You can have autosomal recessive SCID, ADA deficiency, VDAJ recombination deficiency. You can have defects in pre-T cell checkpoint. DiGeorge syndrome is considered SCID. Then there's several others that I won't even cover. So on the X chromosome, we have the gamma chain. I almost said beta chain. We have the common gamma chain for IL-2 type receptors. So you remember that in IL-2 type receptors, there's several of them, but many of them will use a common gamma chain and then a specific chain. I'll just put a specific chain. So this common gamma chain is used in several of the receptors, and then, like say, IL-2 would have its own specific chain, but then it would use the gamma chain as well. And then IL-4 would use its own specific chain, and it would also add the gamma chain to that. And so all of these IL-2 receptors that I have listed here. Now keep in mind there are more IL-2 type receptors. The thing that makes it an IL-2 type receptor is that it uses the JAK-STAT pathway. But the thing that ties all of these together is the fact that they use this common gamma chain in the receptor itself. This gamma chain is made on the X chromosome, and so it's an X-linked skid. And then, so let's see what I've got here. If I can't signal through these mechanisms, if I can't signal through anything using the gamma chain, what will that do? IL-2 receptor, I can't have T cell growth. IL-4 receptor, I can't have B cell class switching to IgE. IL-7, this is the most important one. IL-7 is required for T cell development. IL-7 is produced through the entire process of T cell development, and if you can't signal to the T cell to stay alive, it will die. IL-9, this is an IL-2 independent growth. So IL-2 is growth, but this is another pathway for growth. IL-15, this is the second most important one. Natural killer cell production. Going to the bone marrow and saying, hey, make some natural killer cells. You're not doing that, you're not going to have any natural killer cells. IL-21, this is what allows T cells to go into the Th17 phenotype, and it activates CD8 T cells. So what's going to happen if I don't have any T cells? No T cells means B cells don't get activated, so then you have no B cell response. No T cells means you don't have any of the helper cytokines that T cells produce, so you don't have anything saying, hey, neutrophils, go out and do stuff. Hey, macrophages, go out and do stuff. You don't have that anymore. Adenosine deaminase deficiency. So adenosine gets converted into inosine by adenosine deaminase. 2-deoxyadenosine gets converted into 2-deoxyinosine. These are the irreversible salvage pathways. So if you remember from biochemistry, this is the salvage pathway. It's irreversible reaction to break down these metabolites of ADP, ATP, and so on. And so a failure of these enzymes will increase deoxyadenosine and its precursors. So what are the precursors? Deoxyadenosine triphosphate and S-adenyl something homocysteine. I'll try that again. S-adenosyl homocysteine. So you get these metabolites and the deoxyadenosine itself. These are all toxic and inhibit DNA synthesis. And the developing lymphocytes are less efficient at degrading this stuff already. They're already less efficient at degrading all this stuff, and then you throw it in the hamper of making their adenosine deaminase even worse than it was, and so now they're just going to build up the toxic metabolites and die. People with this usually have deafness, costochondral abnormalities, liver damage, and behavior problems on top of the severe combined immunodeficiency. Another form is this purine nucleoside phosphorylase. It's even more rare, but you get the same phenotype, the same type of idea as you're building up the toxic metabolites in the lymphocytes. The next one is the barelymphocyte syndrome. Barelymphocyte, what happens? You have decreased or absent expression of MHC class II. So you remember the rule of 8, CD4 T cells get activated by MHC class II, CD8 T cells get activated by MHC class 1, 4 times 2 equals 8, 8 times 1 equals 8, so the rule of 8s. And you remember if you have a decreased expression of MHC class 2, then you're not going to activate your CD4 T cells, just isn't going to happen. The other thing is, you're not even going to develop CD4 T cells, so you remember in the development, so you're going to start producing double positive T cells. So these double positive T cells, they're going to express CD4 and CD8 on their surface. And then whenever they get chosen for positive selection, they bind to, so here's my epithelioid cell, and it's going to have MHC2 on it, and it's also going to have MHC1 on it, and depending on which one of these receptors this T cell interacts with will depend on whether or not it turns into a CD4 or a CD8. If it interacts with the MHC class 2, it will develop into the CD4 phenotype. If it interacts with the MHC class 1, it develops into the CD8 phenotype. If I can't present class 2, I don't get CD4 positive T cells. And so again, this is called barelymphocyte syndrome. You get reduced positive selection for CD4 positive T cells. It's the double positive to single positive. In the periphery, you get reduced activation of a reduced population of CD4s. So the few CD4s that you do get, you have a reduced activation of those, and so they don't even work. So also this can happen with CD8s. This can happen with CD8s by defects in TAP. So you'll remember that you have your antigen presenting cell, or not necessarily, you have your MHC1 cell, and what it's doing, so this could be any old cell in the world. This is any cell except for your red blood cell. It's going to have, here's my nucleus, here's my endoplasmic reticulum. Inside this endoplasmic reticulum, it's expressing the MHC2. I'm sorry, the MHC1, I said MHC2. It's expressing the MHC1, and then you're using a TAP protein. TAP protein is pushing cytoplasmic proteins in to be presented on that MHC1. If you have a defect in TAP, it can't push the proteins in, and they won't be expressed on MHC1, and therefore this won't get put to the surface. MHC1 isn't put up to the surface, you don't get selection of CD8 T cells, you don't get any intracellular immunity from the CD8 T cells or anything like that. There's a defect in T cell activation that I think we should be aware of, and I'll put this up here and try to draw this out for you. So you have, of course, you have your TCR. This is my TCR. You have all of your associated proteins. You have your CD4 here. You've got your CD3s, you've got your zeta chains, you've got everything you need to activate this T cell. You get your CD28 and B7, so here's my CD28, and it's combined with B7 from an antigen presenting cell. I've got my MHC2 holding up an antigen. I've got everything going on, and I get down to where I've sent the signal down the chain, and inside my endoplasmic reticulum, I have a bunch of calcium. The calcium flows out and goes and does its signaling. But over here, I have another protein. So that protein is called STIM1, and what it does is it senses the release of all this calcium. When the calcium gets low inside the endoplasmic reticulum, what it does is it will go up to the cell surface, and on the cell surface, I have another protein that's closed up, and it's called crack. I have a crack protein, and it has a small crack in it. And it'll go up to crack, and it'll say open up, and then crack will open up, and calcium will come in, and it will be able to not only help the calcium from the endoplasmic reticulum, but it will also be able to replace the calcium from the endoplasmic reticulum. This crack protein has a subunit in it called ORAE, and when that gets mutated, this extra calcium can't come in, and the CD4 T cell can't be activated. And so let's go back up and see what I wrote really quick. It says a defect in crack reduces influx of extracellular calcium. T cell activation is mediated by calcium signaling. STIM1 senses the ER released from the endoplasmic reticulum, and it will subsequently open crack. It'll open the crack up. So ORAI1, O-R-A-I, however you pronounce that, 1, is a component of crack that is often mutated, causing this defect in T cell activation. Next, we've got antibody deficiencies. So antibody deficiencies from gammaglobulinemia, isolated IgA, CVID, X-linked hyper IgM, autosomal IgMs. And again, the mechanism, BTK, TACI, mutation, CD40s, AID. So we have X-linked agammaglobulinemia, sometimes called Bruton's agammaglobulinemia. And the idea behind this is that we have a B cell that is maturing. And the first thing that B cell does is it goes and it edits its DNA. It comes down with VDJ recombinase, VDJ comes down and starts editing the DNA to make a T cell receptor. And that T cell receptor has two components. It has a heavy chain component and a light chain component. And that heavy chain always gets finished first. He always finishes the race first. So it comes up here, and on the surface I get my heavy chain. And then what the cell does is it puts in a surrogate light chain. There's a surrogate light chain, and it gets all of its links and everything. That's a surrogate. And whenever that is fully expressed, a signal goes down to the nucleus and says, heavy chain's done, start working on light chain now. Heavy chain is done and everything seems to be okay, so go ahead and continue your work. One of the molecules that does the main signaling there is Bruton's tyrosine kinase, or BTK. And if there's a defect in BTK, then you don't get any light chain formed. If BTK is working all right, light chain gets recombined, it gets edited, and it comes up and it replaces the surrogate chain. The surrogate chain is just a dumb chain. It's just standing there making sure everything has structural soundness. And then it gets replaced by the true light chain when everything's done and all the crosslinks get made. And you have a fully working B cell receptor, an IgM or an IgD. But without the BTK, you can't get the T cell to fully mature, so you have no T cells. So that's why it's called gammaglobulinemia, because you can't produce gamma globulins. You can produce immature B cells, but you can't actually produce a mature B cell that makes antibodies. They can't make immunoglobulins. So what I say here, Ig heavy chain gets rearranged, surrogate light chain gets added with it, a signal is induced to start the light chain. That signal's sent by BTK. It doesn't get transduced because BTK is defective. Now, here's the thing. Whenever a baby's born, they've got antibodies in their system from mom. Specifically, they have IgG that can cross the placenta. It crosses the placenta, and it stays in the baby. Now, IgG, the half-life of the various subtypes of IgG change significantly. Most of the texts will give about a 3-week half-life. And I find this confusing because with a 3-week half-life, you would expect the baby to have no IgG at around 2 to 3 months. But they have still significant amounts up to 6 months. This is the classic. This is Robin specifically highlights this in black. It's the classic exam question. A baby, specifically a boy, was sick at 6 months old. That was his first time he got sick. He started getting recurrent infections. You're looking at X-linked gammaglobulinemia. Now, where else do you get Ig from your mom? You get IgA from breast milk. IgA from breast milk to protect the mucosal surfaces. The IgG is actually circulating in the baby's blood. The IgA can only coat the mucosal surfaces, so it doesn't actually go into the child. So it's going to have different phenotypes. If you were not breastfeeding, for example, you would have a higher risk of infection with this disease, but you would still have different phenotypes of, like, if there was no IgG versus not breastfeeding. And I shouldn't say phenotype because we're not talking, not breastfeeding is not a genetic thing. It's just a different clinical picture. It's not nearly as severe. Oh, I was actually going somewhere with that. The reason I was saying that is because we know IgA comes from the breast milk and has some protection, but there was some hearsay from last block that in the initial breast milk, you can get some IgG, and I'm saying that like I don't know if that's right because I can't find it anywhere, but I heard it, but I can't find anything that says it's not true, but I also can't find anything that says that there are not leprechauns dancing on the dark side of the moon. So I mean, just because there's nothing that doesn't say it does not mean that it's a possibility. The answer is I just don't know why the 6-month time period. Now, there are other autosomal recessive, so X-linked recessive, of course, you're going to see it in a male child every time. And the uncles, the mother's brothers are going to have, died at birth as well. So uncles all died at birth, and then this lady had a male child, and he died, or he's sick now or something, at 6 months of age anyhow. But you can have autosomal recessive gammaglobulinemia, and it's the same idea. You have pre-B cell receptor checkpoints to make sure everything is working right, and you have a defect in one of those checkpoint proteins. And when you have a defect, it doesn't allow the cell to move on because it can't say, yep, checkpoint good, go ahead and go to the next stage. It just won't do it. It'll sit there, and it will not mature into a B cell. Okay, isolated IgA, knowing the clinical picture is more important than knowing the molecular biology of this. And I say that because it's almost never determined what the molecular basis is. In the few cases where it is actually determined, they can find BAFF problems, B-cell activating factor, but that's not even the majority of isolated IgA deficiencies. So some sources will call it isolated IgA deficiency, some will call it selective IgA deficiency. Call it whatever you want. I'll just call it IgA deficiency. And of course, IgA protects the mucosal surfaces, so you get mucosal infections, and it's associated strongly with celiac disease. It's associated with an autoimmune disease. And I think that something like 30% of people with celiac disease also have isolated IgA deficiency. And then I think it's something like out of the people that have isolated IgA deficiency, it's like 20%. I really don't remember what the exact numbers are, but it's a high number compared to the general population. So remember, celiac disease is associated with it. Now, common variable immunodeficiency is even worse than isolated IgA in the fact that there are so many mutations with it, none of the textbooks will give you a molecular mechanism. It's probably because there are 50 molecular mechanisms for this. But what you get is defect in T cell, B cell, or both. Could be any of them. Decreased serum immunoglobulins in every case. And so it has a high incidence of autoimmune, not specifically celiac in this case, but you can get like SLE and other things. High association with tumors, especially lymphoma. And 99% of the time it is a diagnosis of exclusion. You excluded everything else, okay, it's CVID. Okay, the next one is hyper-IgM. Hyper-IgM is a misnomer in most cases. Usually you don't get extra IgM. Usually you get reduced Ig everything else. I'll just put all else. All else. And so IgM is high compared to everything else. Now IgM can be a little elevated, but usually it's about normal. So what causes this? Let's go and re-talk about how B cells are activated, or B cells are induced to class switch. So a B cell can be activated in either a T dependent or T independent manner, but only T cells can cause class switching. So you have your B cell right here, and it got its antigen on its B cell receptor, which is probably an IgM, and it internalized it, and now it's expressing it on MHC class 2. So now I had my antigen bind here, I internalized it, now I'm presenting it on MHC class 2. Then I've got my T cell receptor that's saying, yep, I think that's an antigen, I agree with you. So on the T cell, you get that, and you also get the CD, so we got to have B7, which is also the same thing as CD, I think 81 and 86, or maybe it's 80 and 86, but B7, because there's a B7-1 and there's a B7-2, and you're going to have co-stimulation on the CD28. So remember that 7 times CD4, this is CD4, 7 times 4 is 28. So then the CD4 is going to secrete some interleukins. So you can have interleukin 4, you can have interleukin 5, or you can have interferon gamma. So interleukin 4 would induce class switching to IgE. Interleukin 5 would induce class switching to IgA. And interferon would induce class switching to IgG. But in order for that, so that would be like signal. We have this 2 signal thing required. So that'd be like a signal 1. You would also need another signal. So I have another signal on this molecule right here. What is this molecule going to be? This is going to be CD40. CD40 is like, hey, B cell, I'm a T cell. So CD40 is like, hey, I'm a B cell. Where's the T cell at to tell me what to do? And here's the T cell with what's this? This is called CD40 ligand. CD40L, CD40 ligand. So whenever CD40 ligand binds to CD40, you can get class switching with the production of these cytokines. So let's go back and see what was written. So we have B cells can be activated T cell dependent or independent. Only T cell interaction allows isotype switching. So a B cell always produces IgM whenever they first start making antibodies, whenever they first get their VDJ recombination done, they always produce IgM. And then in order to switch to producing something else, they have to have CD40 ligand bind to CD40. And so then, of course, you can have your interleukins and your interferons also telling exactly which of these isotypes you want to pick. Then B cell is active and undergoes somatic mutation. Somatic mutation is done with AID and UNG. So a mutation of CD40 ligand, that's X-linked hyper-IgM. CD40, autosomal recessive hyper-IgM. And then the AID or UNG mutation is an autosomal recessive. And so 2 autosomal recessives. CD40, probably more common than this. CD40 ligand is the most common of the 3. Now we only have a few things left. The next one is, so we got Wiskottaldrich, X-linked lymphoproliferative syndrome, which, remember the question from the beginning, we'll get to that. And then ataxia telangiectasia. Now, with Wiskottaldrich syndrome, you can get it from the Wasp protein, or you can get it from an X-linked WIP protein. We're not going to be covering this because it's the exact same presentation. WIP is a protein that's attached to Wasp and participates in the signaling. So it's the exact same phenotype. It's just a different gene that's mutated. Okay, so we have a protein called the Wasp protein. It stands for Wiskottaldrich Syndrome protein. It is expressed exclusively in bone marrow-derived cells. So here what I've drawn for you is I have a surface receptor, I have a cytoskeleton in red, and then I have this Wiskottaldrich protein that will connect this surface protein to the cytoskeleton. Defect in Wiskottaldrich protein, you get no connection to the cytoskeleton from the surface molecule. So adapters downstream. So what happens? Whenever let's say this was a T cell receptor, whenever the antigen is bound to it, some of the signaling utilizes the cytoskeleton pathways to carry the signal. Not all, not many, but some of them do. And whenever that's defective, there's no attachment, it makes it increasingly hard for them to find the skeleton and send that signal. Also, whenever the signal is sent, you get depolymerization, you get repolymerization, you get like basically a rearrangement of the cytoskeleton that causes movement. And whenever you don't have those receptors connected, you're not going to get the polymerization and the rearrangement to cause that mobility. So you get decreased mobility and decreased activation. A few other associated things is you get thrombocytopenia, and you just think that there's decrease in the cytoskeletal signaling, and so some of the way that the platelets are produced require that intact cytoskeleton. You also can get eczema. I have no idea how. I couldn't find it. And of course, you get defects in cellular immunity, defects in increased repetitive infection. We also have an ATM gene that doesn't stand for money. It's ataxia telangiectasia mutation. So what does ATM do? It actually just sits there and looks at the DNA, says you look pretty, and if there's a break, it doesn't always, but it can activate p53. So what does it do inside of leukocytes? Well, we have this enzyme called VDJ recombinase, and VDJ recombinase goes and you have your DNA, it induces a double-stranded break. So I got a break in my DNA. That's what VDJ recombinase's job is to do. And ATM, ataxia-telangiectasia mutant, whenever it actually stabilizes the VDJ recombinase complex. It stabilizes it, and it also watches to make sure that this gets fixed. If it doesn't get fixed, this and ATM is messed up, this cell doesn't go through apoptosis. It doesn't get killed. And so now you have a bad cell, first of all. And also you have decreased activity of VDJ in the first place because the complex isn't being held together. It's not stable. It can't even do its job of breaking the DNA to edit it, let alone put it back together. Now, so anything that's dividing fast, anything that has the chance of mutating fast, you're thinking of that. So vascular, except for, and I don't know why, but except for skin, probably because there's so many other mechanisms. But you get vascular malformations, you get neurological defects. Neurologic makes a lot of sense to me. Anytime there's a repair enzyme that's missing, it always makes sense to me that you get neurologic defects because there's already a reduced number of mechanisms to repair the DNA because they don't divide. You get increased tumor incidence, so that makes sense. You have breaks in your DNA, p53 is not being activated, so it makes sense. And then immunodeficiency because you're using VDJ recombinase. So I've already hit on the fact that we have to have co-stimulation of T cells, CD28, B7, CD28. This co-stimulation of B cells is required for T cell this co-stimulation of the T cell is required for its activation. But it's only the most well-known form of co-stimulation. There is a CD2 family of proteins that can also co-stimulate, and they have other functions as well. And there's a subset of CD2 known as SLAM, and SLAM stands for sap associated something like it's associated with, I'm sorry, no, I got that wrong. So you have SLAM, and you have SAP, and SAP stands for SLAM associated protein. So you got the SLAM protein, and then you've got this other protein that's associated with it, and we don't really know what to call it, so we're going to call it a SLAM associated protein. So these two proteins can form up this subset of CD2 family receptors, and they're involved in the activation of natural killer cells, T cells, and B cells. They can be alternative co-stimulation pathways, not very well characterized. And the only disease that has been associated with defects in this is Epstein-Barr virus. So you can get fatal mono. There are other clinical features, but as far as an infection, the only infection associated with it is Epstein-Barr virus. So again, this child, he has a primary immunodeficiency. He presented your care with an extremely severe infectious mononucleosis. It can be fatal even. His past medical history was not significant for any other bacterial or viral infections. What is besides Epstein-Barr virus, what is he most likely associated with? It wasn't Wiskottaldrich. It wasn't SCID. It wasn't Bruton's gammaglobulinemia. It wasn't C1 inhibitor. We didn't even talk about complement deficiencies, but you'll know C1 inhibitor is associated with an angioedema. But this SAP and SLAM are X-linked lymphoproliferative syndrome, and they can only be associated with one infection, and that's EBV, so that's the answer. And yes, Dr. McCullough, I did make that up. My question, you can use it any way you want.